Okay, good morning students. In previous class, biotechnology, principles and processes, we are studying regarding the genetic engineering. We are studying regarding the genetic engineering, and there we were discussed regarding the conceptual development of the principles of genetic engineering, and in that we were discussed regarding recombinant DNA, which is done by the gene cloning and gene transfer. Afterwards, we were studying regarding the origin of replication, definition of cloning, plasmid. Okay. Then we were discussed regarding the restriction enzyme, and restriction enzyme, it is also called as a vector, which is helpful for transferring the origin or alien DNA into the host organism. Then we will discuss regarding what genetically modifying an organism, the steps. In that, we will discuss regarding the identification of DNA with the desirable gene, introduction of the identified DNA into the host cell, then maintenance of introduced DNA in the host cell and transfer into its progeny. Okay. After that, in today's class, we used to study tools of recombinant DNA technology. Tools of recombinant DNA technology. What we have discussed in previous class, we have discussed about the different different conceptual development of genetic engineering, principles of genetic engineering. So let us start with the tools of the recombinant DNA technology. In recombinant DNA technology, what are the tools? There are few important tools those are used in a recombinant DNA technology, in the sense genetic engineering. Right? What are those in the sense? First one is restriction enzyme. First one is restriction enzymes. Second one is polymerase enzymes. Third one is ligase. Fourth one is vectors, and next one is host organism. And I told you, and what I explained regarding this in previous class. How we are going to take a restriction enzyme, how we used to cut that, and how we are going to what we are going to call that as a join the cutted chain of DNA with the help of ligase. Then vector, we used to use the plasmid. We were discussed about the plasmid. Then we are going to introduce that host cell, means plasmid into the host organism. These are things we were studying yesterday. So in today's class, tools of recombinant DNA technology. These are the tools of recombinant DNA technology: restriction enzyme, polymerase enzyme, ligase, vector, host organism. Now we used to study in detail regarding the restriction enzyme. Restriction enzyme. What are these restriction enzymes or restriction enzyme? In 1963, two enzymes responsible for restricting in the sense suppress the growth of bacteriophage in Escherichia coli were isolated. Escherichia coli ಇಂದ ಏನು ಮಾಡ್ತಾರೆ ಅಂದಾಗ ಎರಡು enzyme isolate ಮಾಡ್ರು. ಆ enzyme ಏನು ಮಾಡ್ತದೆ ಅಂದಾಗ they are going to suppress or restrict the growth of bacteriophage. What are those amends? How they are going to restrict the growth of bacteriophage? In a sense, there are the two enzymes, and how they are going to play the role in restricting the growth of bacteriophage. One enzyme that is going to go for that enzyme is going to add, or this is going to add a methyl group to the DNA. That is going to add a methyl group to the DNA. Another one enzyme which is helpful for cut the DNA. In such a way that the two enzymes were isolated from the Escherichia coli. Those are going to restrict the growth of bacteriophage, and later on we are going to call these two enzymes are restriction endonuclease. We are going to call them as a restriction endonuclease. Here I written restriction endonuclease. Now, restriction endonuclease. The first restriction endonuclease was named as a HIND2. First restriction endonuclease was named as a HIND2. This is the HIND2 is the first restriction endonuclease. In the sense, here we are going to get one enzyme is used to add a methyl group to the DNA, and another one is cut the DNA. Now here, we cut the DNA in a specific site by recognizing the specific sequence of 6 base pair. 6 base pair. These what hint to what it is going to do in the sense the DNA it has to have what we are going to call that one as a DNA at a specific recognized site in the sense DNA has to have the base pairs in the sense adenine, guanine, cytosine, thymine. Okay, now we ಕೆಲವರು ಆ base pairs ಏನಿರ್ತಾವ ಏನಾಗಿರ್ತಾವ ಅಂತ sequentially join ಆಗಿರ್ತಾವ. sequentially join ಅಂದಾಗ for different amino acids. Okay, ಬೇರೆ ಬೇರೆ ಅಮೈನೋ ಆಸಿಡ್ ಸೀಕ್ರೆಟ್ ಆಗಬೇಕು ಏನಪ್ಪ ಫಾರ್ಮ್ ಆಗಬೇಕು ಅಂತಂದ್ರೆ ಅದು ಡಿಫರೆಂಟ್ ಪ್ರೋಟೀನ್ ಫಾರ್ಮ್ ಆಗಬೇಕಂದ್ರೆ ಸೀಕ್ವೆನ್ಸ್ ಏನಾಗಿರ್ತಾವ ಮತ್ತು ಅರೇಂಜ್ಮೆಂಟ್ ಬೇರೆ ಬೇರೆ ಇರ್ತೈತಿ. ಸೊ ಹಿಯರ್ ವಾಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಹ್ಯಾಪನ್ ಇನ್ ದ ಸೆನ್ಸ್ ಬೇರೆ ಬೇರೆ ಜೀನ್ಸ್ ಸಲುವಾಗಿ ಬೇರೆ ಬೇರೆ ಬೇಸ್ ಪೇರ್ಸ್ ಅರೇಂಜ್ಮೆಂಟ್ ಇರ್ತೈತಿ. ಹಾಗಾಗಿ ನಾನು ಹೇಳ್ತಕ್ಕಂದಾಗ ಹಿಂದ್ ಟು ಇಟ್ ಈಸ್ ಎ ಫಸ್ಟ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ which is to cut the DNA at a specific site by recognizing the specific sequence of 6 base pair. 6 base pair ಅಂತಂದಾಗ 6 base pairs ಇರಬೇಕು. ಅದು ಪರ್ಟಿಕ್ಯುಲರ್ ಯಾವ ರೀತಿ ನಮಗೆ ಏನಪ್ಪ ಅಂದಾಗ ನೆಕ್ಸ್ಟ್ ನಾವು ಏನು ಪ್ರೋಟೀನ್ ಸಿಂಥೆಸಿಸ್ ಮಾಡೋದ್ರಿ ಬೇಸ್ಡ್ ಅಪಾನ್ ದಟ್ it is going to recognize and then it is to cut at a specific site and that specific site is called as a recognizing sequence. ಅದಕ್ಕೆ ಏನಂತ ಕರೀತೀವಿ ನಾವು recognizing sequence ಅಂತ ಕರೀತೀವಿ. Right, so today we have more than 900 of restricted endonuclease. First endonuclease ಯಾವುದು ಹಿಂದ್ ಟು. ಈಗ ಇವತ್ತಿಗೆ means today we have 900 restricted endonuclease from 230 strains of the bacteria. 230 ಬೇರೆ ಬೇರೆ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸ್ಟ್ರೇನ್ ಇಂದ ಇವತ್ತು ನಾವು ಏನು ಐಸೋಲೇಟ್ ಮಾಡಿದೀವಿ ರೆಸ್ಟ್ರಿಕ್ಟೆಡ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ನ ಐಸೋಲೇಟ್ ಮಾಡಿದೀವಿ. ಎಷ್ಟು ರೆಸ್ಟ್ರಿಕ್ಟೆಡ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ನ ಐಸೋಲೇಟ್ ಮಾಡಿರಪ್ಪ ಅಂತಂದಾಗ 900 restricted endonuclease are means isolated from the 230 strains of bacteria. After that what the data ತಂದಾಗ ನಾವು enzyme restricted endonuclease ನಾವು isolate ಮಾಡಾತೀವಿ ಅದಕ್ಕೆ 900 ಅಂತ ಹೇಳಿದ್ವಿ ನಾವು. 900 ನಾವು ಯಾವಾಗ ತೆಗೆದಪ್ಪ ಅಂತ ನಾವು ಎಲ್ಲ ಏನು ಮಾಡಬೇಕು ನೇಮಿಂಗ್ ಮಾಡಬೇಕು. ಯೂಸ್ ಟು ನೇಮ್ ಅಂಡರ್ಸ್ಟುಡ್ ಅವಕ್ಕೆ ಹೆಸರು ಕೊಡಬೇಕು. ಸೊ these restricted endonuclease naming of these enzymes ಹೆಂಗೆ ನಾವು ನೇಮಿಂಗ್ ಮಾಡ್ತೀವಿ. ಫಸ್ಟ್ ನೇಮ್ is a genus. Second 2 letters ಏನು ಬರ್ತಾವ ಅಂತ those are the species of a prokaryotic cell from which they are isolated. ಯಾವ ಒಂದು ಸೆಲ್ ಅದಾಗ ನಾವು ಐಸೋಲೇಟ್ ಮಾಡ್ತೀವಿ ಅವು ಎರಡನೇ 2 ಲೆಟರ್ ಬರ್ತಾವ ಎಲ್ಲಿಂದ ನಾವು ಐಸೋಲೇಟ್ ಮಾಡಿದೀವಿ ಆ ಸೆಲ್ ಹೆಸರು ಕೊಡಬೇಕು. ಅಂಡರ್ಸ್ಟುಡ್, prokaryotic cell. ಫಸ್ಟ್ ನೇಮ್ ಬರೋದು the genus. For example here ECORI. Here this ECORI it is come from Escherichia coli RY13. Escherichia coli RY13. Understood? Now in this what is going to happen, R, whatever the R is there, this R is representing the strain from which we are going to isolate, and Roman, I written here 1, Roman 1, that is the Roman number indicates order in which the enzymes were isolated from the strain. In which order we are going to isolate the enzyme from the strain, that indicates the Roman number. Understood? That indicates the Roman number. That is what how we are going to name the restricted endonuclease. How we are going to name the restricted endonuclease. Now these restricted endonuclease are of 2 types. These restricted endonuclease are of 2 types. What are those in the sense? One is exonuclease, another one is endonuclease. What I told you, EXONUCLEASES, exonuclease and endonuclease. This is the important question. What is mean by restricted endonuclease? Restricted endonuclease are the what we are going to call as molecular seizures. Molecular seizures, and they are having the 2 types of restricted. It means there are the 2 types of restriction endonuclease. One is exonuclease, another one is endonuclease. What is the exonuclease is to do? Exonuclease remove the nucleotides from the end of the DNA. Remove the nucleotides from the end of the DNA. For example, suppose this is the DNA strand and these are the base pairs. Like this something. This is going to remove the end of the DNA. Exonuclease are going to remove the nucleotides from the end of the DNA. End of the DNA. Endonuclease are the restricted endonuclease. Those are may cut at a specific position within the DNA. May cut at a specific position. For example, ಈ ಇಲ್ಲೇ ಬೇಕಂತ position within the DNA. DNA ಒಳಗಡೆ ಏನು ಮಾಡ್ತಾವ ಕಟ್ ಮಾಡ್ತಾವ. That is what endonuclease may cut at a specific position within the DNA. Exonuclease remove the end or nucleotides from the end of the DNA. Remove the nucleotides from the end of the DNA. These are the two kinds of restriction endonuclease. One is exonuclease, another one is endonuclease. Exonuclease, what they are going to do? They are going to remove the nucleotides. What we are going to call that as a from the ends of the DNA. Endonuclease, in the sense, endonuclease may cut at the specific position within the DNA. This is about what? Two kinds of restricted endonuclease. Understood? So many times the question is arise. What are restricted enzymes? Okay, define and write the different kinds of restricted enzymes or restricted endonuclease, exonuclease and endonuclease. What is the difference between exonuclease and endonuclease in the sense? Exonuclease, they are going to remove the nucleotides from the ends of the DNA, and endonuclease is nothing but make cuts at the specific position within the DNA. This is the difference between the endonuclease and exonuclease. Now, next point we are going to study, that is what? Palindromic nucleotide sequence. So here I written ACGT is in the sense. Now in video what we used to call that as the base ಬರ್ಕೋತ ಹೋಗುತ್ತೆ. ಆ base ಒಂದು ಏನಂತೈತಿ ಸೀಕ್ವೆನ್ಸ್ ಇರ್ತೈತಿ. ಆ base ಏನಂತೈತಿ ಸೀಕ್ವೆನ್ಸ್ ಇರ್ತೈತಿ. ಇಲ್ಲಿ ಏನಾಗ್ತೈತಿ ಅಂದಾಗ ಒಂದು ಏನಪ್ಪಾ ಅಂದಾಗ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್ ಬರ್ತೈತಿ. ಆ ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್ಸ್ ನಾವು ಏನು ಕರಿತೀವಿ ಅಂದಾಗ palindromic nucleotide sequence ಅಂತ ಕರಿತೀವಿ. So here what is meant by palindromic in the sense? palindrome ಅಂದ್ರೆ ಏನು? palindrome ಅಂತಂದ್ರೆ ಏನಪ್ಪಾ ಅಂದಾಗ in a particular sequence ಇಲ್ಲಿಂದ ಸಾಲಾಗುತ್ತೆ ಇಟ್ಕೊಂಡು ಇಲ್ಲಿ ಸಮಯಕ್ಕೆ ಪರ್ಟಿಕ್ಯುಲರ್ ಸೀಕ್ವೆನ್ಸ್ ಇಲ್ಲಿಂದ ಇಲ್ಲಿ ಮಟ ಇನ್ನೊಂದೆಲ್ಲ ಕಂಟಿನ್ಯೂ DNA ಈ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಸೀಕ್ವೆನ್ಸ್ ದಾಗ ಏನಾಗುತ್ತೆ palindrome in the sense group of letters when we are going to read those letters from what we are going to call it as a first to last or last to first. In both the direction the letters are same and it is going to give the same meaning. Letters are same and it is going to give the same meaning. That is what we are going to call as a palindrome. Palindrome meaning there are a group of letters. For example here I written on the board Malayalam. It is a group of letters. If you are going to read this from left to right direction or right to left direction, the words are the same and the meaning is also the same. In the sense here, M A L A Y A L A M Malayalam. M A L A Y A L A M Malayalam. In both the sides they are going to repeat the similar letters and it gives the similar meaning. That is what palindrome. Now, in such a way that in particular what we are going to call it as a sequence, we are going to get a particular sequence in a DNA. There the base pairs are arranged in a palindromic manner. Palindromic manner ಅಂತಂದಾಗ ಒಂದು particular position ದಾಗ DNA ದಾಗ base pairs ಏನಾಗಿರ್ತಾವಪ್ಪ ಅಂತಂದ್ರೆ they are arranged in a palindromic manner. Understood? So here I written palindromic nucleotide sequence in the sense palindrome in a DNA is a sequence of a base pair that reads same on a two strands. Two strands of the DNA to double stranded DNA ಇರ್ತೈತಿ. ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಇರ್ತವೆ. ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಮೇಲೆ ಏನಾಗಿತ್ತಪ್ಪ ಅಂದಾಗ ಒಂದೇ ರೀತಿ ಅದನ್ನ ನಾವು ಓದ್ತೀವಿ. ಒಂದೇ ರೀತಿ ನೀವು ಮುಂದಿನ ಹಿಂದ್ಗಡೆ ಅಥವಾ ಲೆಫ್ಟ್ ಸೈಡ್ ಇಂದ ರೈಟ್ ಸೈಡ್ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇಲ್ಲಿ ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಅಂತ ನೀವು ಹೇಳ್ತೀನಿ ನಾನು. I should read here. First, palindrome in the DNA is a sequence of a base pair that reads same on the two strands when orientation of a reading is kept same. Orientation of a reading is kept same ಅಂತಂದಾಗ ಏನು ನೋಡೋಣ ಈ ಕಡೆ ಹೋದ್ರೆ Malayalam ಈ ಕಡೆ ಹೋದ್ರೆ Malayalam. Understood? In such a way that in cDNA, the base pairs are arranged in a palindromic manner. That's what when we are going to read in a both stand in a both direction, the base pairs are gave the same orientation or a same orientation of reading is kept the same. Understood? So here I can see 5n to 3n, 3n to 5n. You know GAA TTC. You can know that CCT AAG. In such a way that the palindromic, what we are going to call it as a sequences, are going to read from in both the what we are going to call it as a strands in a similar manner. Understood? GAA TTC and third to fifth CCT AAG. This is about what? Palindromic sequence. This is about the palindromic sequence, and this is also the question is going to arise. What is mean by palindromic nucleotide sequence? That time you are going to write the definition of this, and then you are going to give the what we are going to call it as a example for the palindromic sequence. Palindromic nucleotide sequence. Okay, so this is about today's class. In today's class, what else we are discussing in the sense we are studying regarding the restriction enzyme, and after that restriction enzyme, then what are the two enzymes, what they used to do. One enzyme is added the methyl group to the DNA, one enzyme is going to cut the DNA at a specific site, and later on these two enzymes are called as a restriction endonuclease. The first restriction endonuclease is in the 2. First restriction endonuclease is in the 2. That is going to cut the DNA at a specific site by recognizing the specific sequence of a 6 base pairs, and that is what we are going to call that as a recognizing site. Then next, today we have more than 900 of restricted enzymes from the 230 strains of bacteria, and how we are going to name these means how they are named these 900 of strains, more than 900 of restricted endonuclease. Naming of enzymes, first name is genus, second two letters are the species, the prokaryotic cell from which they are isolated. The next example, E. coli, Listeria coli, RY13, and in this R is a strain and Y is representing the Roman number, that order in which the enzymes were isolated from the strain. That is very important. And afterwards we will study regarding the two kinds of restriction endonuclease. What are those in the sense? Exonuclease, endonuclease. Exonuclease in the sense these are the restricted endonuclease which are going to remove the nucleotides from the ends of the DNA. Endonuclease, they are going to make cut at the specific position within the DNA. They are going to make cut at the specific position within the DNA as shown here. Then we will discuss regarding palindromic nucleotide sequence. In palindromic nucleotide sequence, what is palindrome in the sense? A group of letter when we are going to read that from left to right or right to left, or from one end to the another end and opposite from next end to the again the first end, that time the letters are same and the orientation of reading is also same or meaning is also same. For example, like given you Malayalam, we used to write these things in exam. What is palindromic nucleotide sequence in the sense? Palindrome on a DNA is a sequence of a base pairs that reads same on the two strands when orientation of reading is kept the same. Orientation of reading is kept the same. GAA TTC 5 ರಿಂದ 3 ತನಕ 5 ರಿಂದ ನೀವು ಮುಂದೆ ಹೋದಾಗ ಮತ್ತೆ GAA TTC. This is about what palindromic sequence. Likewise, we are going to remember these things regarding the restriction endonuclease. Important thing is first restricted endonuclease is in the restricted endonuclease, and nowadays more than 900 restricted endonuclease were isolated from the 230 strains of bacteria. And how we are going to name the restricted enzymes. Afterwards, we are going to remember the difference between the exonuclease and endonuclease, then palindromic nucleotide sequence, definition and example. Right? Then in the next class, I am going to tell you about the diagrammatic representation of recombinant DNA technology. The recombinant DNA technology is the diagrammatic representation. Then we are going to study about next thing, that is the separation and isolation of DNA fragment. Separation and isolation of DNA fragment. And afterwards we used to study these things also, polymerase enzyme, ligase, vectors, host organisms, etc. So this is the today's class. Here we were discussed about the restriction enzyme. Okay, with this I am going to stop here, and in next class I am going to start with the diagrammatic representation of recombinant DNA technology. So you are going to remember these things: restricted endonuclease, first restricted endonuclease, two kinds of endonuclease, exonuclease and endonuclease, polyandromic nucleotide sequence. These are very important as a examination point of view. Okay, I am going to stop here. Thank you. We continue next.